Okay. So what were our topics again? I'm sorry. I'm totally focused now.
Genetics and DNA, cell communication, and DNA history.
Got it. The history of the DNA particles. Um, awesome. I think let's let's go with some genetics. Let's go with genetics, because that's a pretty good topic we could cover. We can make some good headway in it. This actually is going to start with chapter 14, and it's going to cover a little bit of 15 for our focus for genetics. Definitely want to make sure we're all like not texting today. When we're doing reviews, this is like it's a non-texting day. If I see your phone, I'm just going to put it somewhere else. Probably the principal's desk. She can deal with it. Sorry. But I think the first thing to talk about with genetics is like a whodunit. So who's our guy? So Mendel. And what did he study? Yeah, his little baby pea plants. His little baby pea plants. And what did he do with his little baby pea plants? You know, he crossed them, right? Now, not only was he having like little baby pea plant A and little baby pea plant B that happened to be a boy and a girl. Not only was he like mating them, but then they were mating each other, right? I mean, there was like this self-fertilization, this cross-fertilization. So like he was basically he was setting this up, if you will. And not only was he setting this up, but he was able to control the setups, or the hookups, if you will. But he was also like taking vast amount of numbers at the same time too. So tons of numbers. Some of the characters that he described or that he studied was like plant height, correct? Seed color, seed shape, was it round or wrinkled, and a slew of other things. But I want to focus our efforts right now on probably like just tallness. We'll use that as an example. Tallness, cool. And the first thing I want to talk about is what does it mean to be true breeding? So what is a true breeding species or a member genetically? What is it? What does that mean? Yeah, you got to be homozygous. So true breeding is basically the same thing as saying homozygous. Is he awake? Julio. Is he taking notes? Would you ask him to take notes, please? Good. So, because it's homozygous, it's able to produce like offspring that are the exact same as the parent, and it's a guarantee. So, for instance, if you have big T crossed with big T, those are both true breeding. Correct? What would all of their offspring be? All their offspring would be tall. What if a big T self-fertilized? What would all of its offspring be? That would be the same as crossing with big T, right? If it was to self-fertilize. And so all of its offspring would be big T. That's what true breeding would mean. As opposed to like a not true breeding plant. Like for instance, your big T, little t, your heterozygous would be considered to be not true breeding. Which means that there's not a guarantee that it can produce offspring that are identical to the parent. This parent right here, if it was big T, little t, is going to obviously be tall. But if it was to fertilize with itself, we can see down here that there would be a quarter chance that their offspring would be short. That's why it's not true breeding. So because of these experiments, Mendel was able to come up with this concept that you had members that were homozygous dominant, homozygous recessive, and then heterozygous. So homozygous recessive, homozygous dominant. Is he awake? Is he taking notes? Leo, would you get him to take notes, please? Please? No? Okay. Why? So, heterozygous would mean that you have two unique characteristics. Homozygous means you have two similar characteristics, right? And remember, this is like what's embedded in your DNA. Now, what would I call what is the T?
inside today, man?
No, man.
I'm sick, dude.
I'm sick too.
I'm sick of
Probably. I don't know. Maybe.
Yeah.
I'm sorry.
It's mental notes today. When I'm sick and I'm not on function, but I just absorb it. I don't need to take notes.
So, where was I at?
I think alleles.
That's where I was going. Alleles. Big T versus little t. What are they coding for? They're coding for tallness, correct? But big T codes for tallness and little t codes for not being tall, right? We don't want to say it codes for shortness. It doesn't code for tallness. Like, the reason that the plant is short is because it doesn't have the tall gene. Not because it has a short gene. It just has a gene that doesn't turn on the tall protein. Does that make sense? So it's either you're having the tall protein or you're not having the tall protein. It's not that you're having a tall protein and a short protein. It's either there's just like the protein for tallness there or absent. No. So, alleles. That would be like the different versions of a gene, correct? Now, if you look at, and I think we probably have our the crosses are okay, right? Like, for instance, if I was to show you, before I talk about some unique characteristics, if I was to do this, and you were to do this cross right here, like, you're okay with saying that that's like a 1-to-1 genotypic ratio, but a 4-to-0 or 100% phenotypic ratio? Y'all cool with that? That's 100% totally fair. What if I crossed something like this? It makes no sense. Cool. And what if I want to know the shot or the percent chance of getting a tall, and let's say that big A was coding for blue, so a tall blue, whatever that might be, and where blue was coded by big A, or big A, little a. So you can do these crosses in your head, correct? What's the chance of the progeny being tall? 100%, right? 4 out of 4. You can do the big T, little t with big T. You get 4 out of 4 guarantee. What's the chance of your plants being big A, or big A, little a, which codes for blue? 100%, right? Cross big A with little a, and you can see that they're all going to be big A, little a, so they're all going to be blue. So that's going to be another 100%. Y'all remember how, so instead of doing like a 16 box, you do a bunch of 4 boxes and you multiply them, you got it made? We want to see another problem like that, are we cool? That's fine. Then there's always, like, another favorite question would be like this one. How many different types of, let's say that these were your genes. Big A, little a, big B, little c. So like from mom, from dad, from mom, dad, whatever. How many different like gamete cells can you make? How many different gamete cells could you make with this setup? How many? 4. 2 times 1 times 1 times 2. 4. Make sense? So how many different gamete cells could you make maybe with this setup? That's Q's at the end. How many? Perfect, 8. 2 times 2 times 2 times 1 times 1. Yay? Awesome sauce. Me? Oh yeah. You want to talk about that one too?
Yeah.
Hardy-Weinberg equilibriums. That's a good topic too. So I think these types of genetics problems are okay, right? And then like reading them and like being able to see, like, it says that the little mouse has like brown is dominant to black, or or whatever like that. Bless you, by the way. I'm sure those are totally cool. Like just reading a genetics problem instead of just getting little A's and little B's. Then there's like the unique genetic vocab that you have to focus on. And some of that vocab would include like the terms epistasis, polygenic inheritance, pleiotropy, incomplete dominance, and then multiple alleles. Cool. Awesome. So can we talk about like what epistasis is real quick? What is epistasis? Y'all remember? This everyone, like when I try to make little jokes about my life with these, everyone loves this one the most.
Oh, yeah.
This is the I go home, the thermostat set on 68. I realize that we got bills. I turn it back up to 76. What does she do? Turns it down to 68. We argue, we fight. Horrible situation. I could solve this very quickly by doing what? I flip the breaker. So she thinks, she sets it on 68. She doesn't realize it doesn't come on. We're all good.
Did that really happen?
Never heard
Just making sure.
Never heard that?
that. No, tell us that story.
That's a good one.
No,
Tells
talk
it
about
like
Sorry, it's 3 classes. Hard to remember sometimes. Sorry. I have one. She has a control spot, and then I control whether her control spot even is on. Right? One gene controls the expression of another gene. Do y'all remember going to the Sam Rhine's genetics concert a couple of months back? One thing he said was like that, like his entire theme is like this concept of like new genetics or epigenetics, and that's kind of why he does those little concerts throughout the nation, is like so he can like talk about epistasis and epigenetics. Polygenic inheritance. What's this?
Wait, can you give me an example of
Epistasis. The one your book gives is just cute enough. They give like mouse color. The mouse can either be like black or brown depending on if it's like big B, little b, or little b. And then there's another color like for C, or there's a gene called C gene that controls whether or not the color is expressed. So like if your little mouse is little c with big B, little b, well, that mouse should be black based on that setup, but since it's little c, it doesn't display color, so it's actually just a white mouse. Or if you had a mouse that was big C with big B, what color would this mouse be? Brown, black, or white? This one would be black, because that's coding for black, and this gene is signifying that the B gene is on. So this is kind of like, this is her thermostat, and we both kind of control it with our little B's and big B's, and my gene is the one that controls whether or not the thermostat's expressed. My breaker is epistatic, if you will, to her thermostat. Cool. Polygenic inheritance. What is this? Please remember. Perfect. Multiple genes. Code for one trait. Give me some examples. Hair color to some degree. Human tallness. And skin color. What? Both types of multiple alleles. Multiple allele system. It's anything where, like, when you look at our differences, if our differences have a very graded or a very how do you say this? Like a very, very, I guess, like it's not definitive, I guess. Like earlobes. Here's one. Earlobes. They're either attached or they're free, right? Your thumb. It's either like pretty straight or it's like it bends back a lot and you have what's called a hitchhiker's thumb. Makes sense? That's either like one option or the other. Or for like the little mice, they're either black or brown or white. It's one or the other. But like our skin colors have different tones, right? Is there just like a group of 5'11" people and then 5'6" people on this planet? There's like people of a unique height that are very like I guess varied, if you will, right? That would mean that there's got to be many genes controlling those features. When there's a graded trait between the individuals of a population, it's got to be that there's many genes controlling that one trait. Skin color, height are good examples. Question. Pleiotropy. What does this mean?
So, one gene affects different traits. Right?
Perfect.
Really?
Yes.
But they're only physical traits right now.
Yes. That's correct. What was the example, or what is an example? Because there's a slew of them around there. That you have one gene that has many effects. We could be cute. We could say like the cool gene, which I obviously have, right? I mean, like I'm a baller, DJ. I can put together a computer. I got a pool.
If you DJ for prom, you have that gene.
I guess that means I won't have that gene. They haven't asked me now. So I guess that would mean that like that's one gene and many effects, right? Now that's a cute one, but let's do a sad one real quick. Well, give me an example of like a gene that is an issue that has many effects. Cystic fibrosis, that's one. One little gene's messed up, you see a slew of physical effects. Sickle cell anemia, that's another one. Bad gene codes for bad hemoglobin, many physical effects though. What? You could go about anything, any kind of physical like impairment. Even nowadays, we could say cancer because we believe cancer is more genetic than it's not. You know, it is certainly more genetic now than we have thought in prior years. Even that would be like pleiotropy, because obviously a cancer piece or a cancerous like segment of DNA, obviously is having like more than just like the one outcome. Then like there's obviously a slew of things that are affected from the cancer DNA. Incomplete dominance. Which one was this one? I need you to think about one thing when you think of incomplete dominance. The flowers. I need you to think of the snapdragons. If you remember the snapdragons, it was big R was red, little r was white, and if Mendel was having it his way, isn't that like McDonald's? Have you? Oh man. Well, anyway, if it was really crazy. It was big R, little r. Mendel would have suspected it would be red, but actually, the heterozygous is pink. And this is the only time where actually the blending hypothesis that your book mentions in chapter 14 is 100% true. Blending hypothesis happens with incomplete dominance, which is a feature or an inheritance model that's not very common. Where the heterozygous is the intermediate trait. Multiple alleles. What's this? That's like blood types. Yep. Remember Mendel suspected that there was just big T or little t, right? Do you ever see like Mendel mentioning like there's big T, there's little t, then there's medium-sized t? Did you ever get that? But in multiple alleles, we see that there's more than just two forms of the gene. How many different forms are there for the blood types? Careful. 3. There's I, IA, and IB. Mendel proposed how many genes were there maximum for any given trait? 2. He thought there was big T, little t, or big B, little b, or big V, little v. Whatever there's out there, he thought there was the big and the small, and that was it. He thought there was a dominant and recessive. End of discussion. But we've learned that in some cases, there's more than 2 alleles out there. Now question, how many alleles do you have max? Like for blood types. You got max of 2, because you got one from the mommy and one from the daddy. I'm type AB blood. I have this allele and this allele. Do I have the little I allele? No, dude. I obviously don't. Now, what if I was to tell you that like I had AB blood, but my dad was type O? Hey, you wouldn't be my dad. Are you able to do that very quickly in your head? Like, see how that is? If I'm type AB, that means, okay, I obviously got an A or a B from the dad and an A or B from the mom. And if my dad was type O, he would have 2 little baby eyes, and he couldn't have given me those 2 things. So let's remind ourselves. II goes for O blood. IAI or IAIA codes for A blood. IBI or IBIB codes for B blood. And let's see, IAIB codes for AB blood. Pretty logical like distinction, I think, between everything. Me? Yeah, I mean, I can't think of any, but there are. There's got to be, obviously. But I sadly like do not know of them. And it's not that like they're like these hidden traits. Like it's gonna be something like simple, like I don't know, the shape of your nostril, or something petty like that. It's just something that's obviously like your body's having, but I can't think of a gene that is following that logic. If they ask you about incomplete dominance from a perspective of an application, it's going to be with blood types and only blood types. So, I think we understand like blood type stuff pretty well. As far as like the different like ways that this vocab could be asked, typically it'll be asked like one of two ways. One is in an application setting. The other way is going to be like in a setting of like knowing the differences between them, like in a little matching section. So that's pretty common as well. Then I heard someone say, what about this Hardy-Weinberg stuff? Let's do Hardy-Weinberg. So, do you remember what Hardy-Weinberg equilibrium was from back in the day? The equation. That equation. That was one equation, and the other equation was... Awesome. Now, what did the P stand for? The dominant allele. And what did Q stand for? The recessive allele, right? Cool. This is P squared, right? In math, what's another way to write P squared? So little p and little p next to each other. So if P was for the homozygous, like, or the dominant allele, P squared would be for homozygous dominance. What would the Q squared be for? Homozygous recessive. Cool. And what would the 2PQ part be for? Now, everyone's like, why is there 2 there? Well, the best thing I could help you with is thinking about Mendel's like major cross. It looks like that. You have 1 big T, coefficient 1 there, 1 little t, and 2 that are big T, little t. Mm-hmm. Yep. Just the way the math could work. Yay? Now, if I said, and let's go back, let's pretend, let's just say that you were Mendel. Let's say you're a Mendel. Let's say that was, let's just say you saw a tall pea plant. You see a tall pea plant. Okay. Does that mean that it's big T? Does that mean it's big T, little t? Could be either, right? You see a tall pea plant, right, Amy? It could be big T, or big T, little t, correct? Could you determine the frequency of a P or a Q of a dominant or a recessive allele based on what that parent is? But here's what you could do. You could find out the number of people in your population, and this is the trick to it. Like if you were the I don't know Hardy-Weinberg equilibrium, this next like 1 minute is the solution to it. If you have a population of humans or of whatever, and if you know the number of members that have the dominant issue or have the dominant gene, you can't do anything Hardy-Weinberg to it. What you have to know is you have to know the number of people that have the recessive condition. Let's say, as an example, let's just say for an example, that we were in a condition or in a world where 16% of the population had cystic fibrosis, which is well above what it is in reality. Cystic fibrosis is a homozygous recessive condition. So if you have 16% of the population that has CF or cystic fibrosis, what does that mean in terms of T? Does that mean 16% are big T, big T, big T, little t, or little t, little t? Yeah, that's this guy, right? 16% are that one, right? Which is going to be which one of these values? So that means my Q squared is equal to 0.16. Now you can do some simple math. How would you solve for the variable mathematically here? Square root of what? Of both sides, right? So what is that root? 0.4? Q is equal to 0.4? What does that mean that P would be equal to? 0.6, right? Just because P plus Q is 1, so it's a simple subtraction. That means that out of the, if you look at the entire gene pool based on these percentages, 40% of the of all alleles in that gene pool. Remember, how many alleles do you have, by the way? 2. One from the mommy, one from the daddy. Could they be the same? Of course. You could get 2 big T's from both parents, but maybe it's 2 little T's. But I'm saying that 40% of all alleles out in that gene pool, according to this stat, is going to be the little t, and 60% is going to be the big T allele. Now, the power of this is, let's say that you have cystic fibrosis, and you fall in the 16%. You would know you would have it, correct? Because there's conditions that associate themselves. So what would be the use of this data? Perfect. Carriers. You could do some math if you wanted to. What's the chance that I'm a carrier? What's the chance that my spouse is a carrier? And what's the chance of therefore our kid having this issue? Make sense? If I look at 2 times P times Q, which is 2 times 0.6 times 0.4, what do I get? 0.48, which is 48%. That means there's a 48% chance that you're a carrier, or that there's a 48% prevalence of carriers in this population. Looking at this, the rest of these stats, it would make sense there's like 36%, which is Q squared or P squared, that are going to be homozygous dominant for the issue, which means 36% of the people in the population not only do not have cystic fibrosis, but they do not carry cystic fibrosis. Now, I told you the 16% was way too high, correct? It would make sense that it's like not high at all. Is cystic fibrosis a positive or a negative? It could lead to the end, right? What would evolution do with that? Would it keep it around? I mean, evolution is like Survivor, like the CBS show. There's the removal of that which is bad. So like evolution would work to remove it. So actually this percent's like 0.0001 or something like that percent, or 0.1, a very small amount. Give yourself like what, maybe another 4,000 or 5,000 years, this value should become nothing. Well, actually, it probably will go up because we're evolving, or we're increasing technology. But in a non-technological world, the value or the percentage of a negative condition should always go down. Question, Sarah?
What was the name of the muscle degeneration that happened around like the 30s or something?
Yeah, okay. Huntington's disease. And it typically, Huntington's disease, she asked, what's the condition that affects like people when they're older, and that's Huntington's disease. Remember, it typically affects males. And what happens is the males are, okay, obviously they are affected, duh. But what else happens to them is they end up like having babies and passing on the trait, or at least the gene onto their children before they realize that they have the issue. In other words, to not to make anyone sad or whatever, but I could possibly have Huntington's disease. Would I know it? No. I mean, well, probably, because there would be a genetic test probably about the time I was a baby, but let's just say that there wasn't. Then I wouldn't know it. When would I know it? I'm 45 years old or so. I have it the entire time, but it doesn't manifest. It doesn't become an issue till I'm older, right? That means that gene that has caused what would be the end of my life within about 5 years of recognizing that I have the issue, I would pass on to my kids. I never know it. That's the only way that you can escape like evolution's like fix of removal of a bad gene, is for the gene to wait till post reproduction to manifest or to display. Which is why Huntington's is still a topic, because if you get this, any disease that is genetic that currently exists, you'll notice that very few people get it, right? Evolution has worked on that number to make it so small. It will continue to work on that number to make it even smaller. And every genetic condition that exists out there, if it was high at one point in time, has been minimized today. If you become a physician, or in other words a doctor, when you grow up, you will see maybe one person in your career that has Huntington's. One person, and if you're just a general family practitioner, one person that has cystic fibrosis and one person that has this issue, because they're not very prevalent. Because evolution has worked to remove them. They're negative. Good? Awesome. I know with the genetics part, we still have